research scholars and the people from industry. On behalf of Department of Pharmaceutical Science and Technology, Maulana Abul Kalam Azad University of Technology, we heartily welcome all the panelists today and our beloved faculty members, dear research scholars and industry people. Today is the day 2 of the FDP program, which is organized by our department, MAKAUT. Today is the technical session 2. Now, today we have with us Professor Pranabis Chakraborty. He is the Director of Food, Pharmaceutical and Medical Science of Technology School, Maulana Abul Kalam Azad University of Technology. We have also in our panel Professor Dr. Jayanta Chattopadhyay. He is also a stalwart person with huge industrial experience. He is currently working as a professor in the Department of Pharmaceutical Science and Technology, MAKAUT. He will basically today give summary of the presentation and give concluding remarks. Today, the eminent speaker is Mr. Sampad Bhattacharya. So let me introduce Mr. Sampad Bhattacharya with you. He is a stalwart person with huge international experience and international importance. He is an alumni of Jadavpur University, Department of Pharmaceutical Science, Department of Pharmaceutical Technology, 1979 batch. He has been trained in productivity and quality management in Japan under UNIDO fellowship and with All India Management Association. Mr. Sampad Bhattacharya has formal research experience in process engineering and scale-up. He is associated with pharmaceutical industry for about 40 years in various capacity and function, including manufacturing, quality control, quality assurance, regulatory affairs, pharmaceutical development in companies like Dufer Interferon, Hindustan Antibiotics, MJ Pharmaceuticals, Alembic, and Sun Pharmaceutical. Mr. Bhattacharya has played significant role in technology transfer, facility and system development, and regulatory interaction in various Indian and overseas projects. He has retired in 2015 from Sun Pharmaceuticals as a Senior Vice President, Sun Global Operation, and Head of the Manufacturing Science and Technology, MSNT. Post-retirement, Mr. Bhattacharya was retained by Sun Pharmaceuticals again as a full-time consultant for developing technical leadership. Now he works as a consultant and pharmaceutical industry expert with Boston Consultancy Group, BCG, in the field of productivity management, quality management, compliance, technical troubleshooting, facility, and systems audit. And also he is involved in designing evaluation, technical competency, mapping, and skill development. He has flair of teaching and mentoring, and he remained associated with Gujarat Technical University, MS University, AICT, BITS Pilani, many other renowned pharmacy educational institutions in India and abroad, in curriculum designing and also a visiting faculty and examiner. He has a number of international granted patents and publication in international journal to credit. So it's a really immense pleasure to introduce Mr. Bhattacharya to our participants. He's a really great personality with international importance. So without wasting much time, I would request Mr. Sampad Bhattacharya to start his presentation. Sir, please.
Do I take the presentation on right way?
Right.
Or there is somebody else to talk in between?
No.
Okay, fine. Thank you. Good evening, everybody. Let me start by a small anecdote. The story everybody is aware of, which may be relevant to make a beginning in the subject. All of us might have, rather we know the story of Sabari, and the story goes like that: when Bhagavan Ramchandra came to meet her, the great fan of Ramchandra being Sabari, she wanted to offer her the plums, what she collected from some trees or something like that. But she wanted to ensure that none of the pieces should be sour. So what she did is she had tasted every piece of the plum, every unit of plum rather in technical language, by little biting, and then collected the heap. And when she was about to offer, then there was not a single piece which was not half eaten or something like that. Of course, the story ends like that. The Bhagavan Ramchandra then accepts everything out of love and affection and respect for Sabri. But let us translate that thing to pharmaceutical manufacturing field or pharmaceutical industry rather. Can we afford to test every piece or rather every unit of our product, and by that ensure that it meets the expected specification or meets the specification or expectation, and that's how we assure that the ultimate quality, expected quality is met with. It seems little unrealistic, but what is the solution to that? We invented statistics and statistical sampling theories, by which acceptance sampling, we draw some sample and assume that it represents the universe, and also assume a principle of degree of homogeneity and the principle of confidence interval. And that's how we assume that the sample what we draw, it represents, it may be scientifically drawn sample, and it represents the every part of the population. And then what the sample is drawn, and from that maybe 20, 30, 40, whatever tablets or capsules are taken for testing, that goes for testing. And it's a matter of chance that those units which come under testing hammer, if they meet the specification, and finally will declare that the batch passes or not, and that decides the fate of the batch. Are we very satisfied and there is no gap in that assumption? If there is no gap in that assumption, then how the products fail? The products fail not only in the within the boundaries of the factory, rather it also fails when it is released. Sometimes it is released by the all the testing that has been done, and it goes to market, and that is how we have incidences like that. This is a very not too very old example. The ritonavir is Abbott's product. When launched in the market in 2012, after launching, it was found that the sum of the units were or the product as was a anhydrous material filled into capsule. The product turned into crystalline, and it failed in the dissolution testing, and it meets the acceptance criteria. Of course, it does not meet the acceptance criteria when tested in the field, and it has to be withdrawn, redesigned, reformulated, and triggered new FDA submission. And you can understand the cost implication. This is just an example after launching, but products also fail regularly in the quality control, quality assurance scenario within the factory background. And let us now understand the failure economics. Average batch failure rate in industry, I'm telling of the global average, is about 2%, between 2 to 3%. In sterile, it may be 3. In general benchmark is around 2. In the non-sterile, around 2% of the batches it fails. That means 2% of the revenue it fails. An average manufacturing cost is 27% of the revenue. Then What is the revenue? Let us take 2019-20 figure, $1.3 trillion US. And if we do the math, simply multiplication of the 27% of 1.3 trillion and multiplied by 2% of it, it is around $7 billion US. Now, this is without considering the process losses, manufacturing efficiency loss, yield losses, which is around 5 to 10%, depending on the technology and the dosage form of the manufacturing cost. And if we add that, then it works out roughly the value loss is to the tune of around 40 billion US. Now, it is been estimated by the figures volunteered by the top 30 pharma companies of the world that they alone can save, with a very conservative figure, $12 billion on such account. So the size of the opportunity to prevention of failure and such the consequent loss is huge. Do you know what is the amount of investment today since 2019-20 in R&D globally is around $190 billion. So assume around $40 billion is an opportunity in a year simply by preventing the failures, then it is what's making investment. Now let us proceed into why the products fail. The general notion is the product fail is caused by many operational and human factors, including human error and all these things, wear and tear, variability of raw materials, active ingredients, unclear procedure, not following work instruction, etc., etc. Although the above mentioned notions are relatively true and true to some extent, the reality is that the variability primarily arises from inadequate design of the product and the process and equipment and its understanding. And process understanding, the mapping of the process quality risk, and is the key to the prevention of failure. The process understanding, if you want to understand, is the knowledge that allows the products to be consistently and robustly meet the standard quality requirement by addressing the design aspect of it and the risk factors that we all have read in our books and textbooks. Now let us understand a little bit about what is the difference between the product and process development. Product development, in very simple terms, is aims to improve the product characteristics, whereas process development, since today's caption of the presentation is process development, let me talk about a little bit about process development. It aims at improving the efficiency of the production process. That means quality, consistency, and productivity. Although it is said that the quality is actually as good as is the quality of design of a product, but the process itself actually contributes to the product development. Quite often, as I'm going to the product development activity and the process development activity improvement, and they are well integrated. That means process development moves along with the product development. And the process development project can thus be an opportunity for the product development, and the product development opportunity can be combined with the process development and cost reduction in production processes. Again, process development explained is like that. It basically starts with a characterization of the process feed material, and then a conceptual flow sheet is drawn to assess the quality and efficiency objectives, and initial laboratory experiments are conducted, and then it focuses on refining the core process workflow or reaction. And from this point of view, it is a cyclical process, rather iterative process, and where the flow sheet is redrawn and the process criteria are listed. The process development of our options and approach is based on knowledge and understanding derived from a series of measurements. I'm highlighting the word measurements and the interpretation of the relationship between the key variables in materials and process conditions. It's just an example of the solid dosage workflow. If we see generically a solid dosage, say operation or unit operation activities, or very generically, what is it is? Basically, dispersion, irrespective of what is the solid dosage, it's a dispersion, densification, discretization, distribution, and dosing is generic definition of a solid oral product development. And therefore, each step, there are so many process options, or there are unit operand process and unit operation options. The dispersion may be co-shifting by co-milling, mass mixing, solvent dispersion, by so many methods. The densification may be high shear wet granulation, your fluid bed spraying, then maybe granulation, wet granulation, so many kind of things, melt granulation, co-extrusion, and so on. I'm not explaining, you can read and understand very simple things. But how these options are made? These options are made based on certain measurements and certain experimentation. While developing process, what are the typical the question it comes for during the process development case? If you want to monitor, want to measure something, what are the typical question? Very layman's question. How does the homogeneity of the blend change with time and geometry? When does the wet granulation reach an end point with reference to the water addition rate? How the particle morphology, size, shape, density, effect, and the flow property, content, uniformity of solid dosage form with final drug release rate is correlate with. How the granulation, compression, coating we are affected by scales. How the force compaction and the roller compaction is related to the granulation morphology, size, and drug release rate. How the coating thickness is built. How it impacts the surface integrity, drug release rate. When does a product form? And at what rate? This is for the chemical reactions or the API manufacturing. When does the reactants disappear? Does the reaction proceed via reactive intermediate? How the impurity is generated at what rate? Whether factors and conditions for them can be identified and controlled. When does the crystallization start and what factors control the rate? What polymorphic forms occur during processing? How the drying rate of lyophilization, water content, lyophilization cycle progress, how the cake structure of the lyophilized product is influenced, how can it be optimized? So these are examples and not limited to this list. So these are the common logical questions and worries when a process development process is initiated. And process development approaches, generally the traditional approach relied upon the process monitoring through static sampling, measurement, and monitoring.
Sir, kindly share your screen, sir. Screen is not visible, sir.
Is it not?
No, sir, your screen is not visible, sir.
Please, you should have told me earlier. Was it visible?
No, sir. Kindly share it, sir.
I'll share it, definitely. But is it visible now?
No, sir. Sir, at the bottom, sir, behind the mute start video.
I know that.
After clicking the main screen, at the right bottom, there will be a button share. That you have to click, sir.
I know that. It happens every time. I don't know why it's not happening this time. Let me see.
Yes, sir. So after selecting the applications,
your video is there. Your share screen is on.
You have to select that particular topic from share screen. You have to select that file.
Yes, so you have to select that application, and after that, at the right bottom, there is share screen button. You have to just press on it, sir.
নিচে যে শেয়ার স্ক্রিন আছে না একদম নিচে ওটা ক্লিক করুন হ্যাঁ ক্লিক করেছেন তো এবার কি শো করছে
showing whiteboard basic advanced file phone pad
এবার ওই যে আপনার পাওয়ার পয়েন্টটা দেখাচ্ছে ওখানে কোথাও দেখুন
it is being shown
হ্যাঁ এবার দেখুন
I think this version is something
perfect এবার চলে আসবে
right
perfect এবার ঠিক আছে
okay
thank you
this I have to share your presentation mode, otherwise you will not get the did you get the presentation mode?
This is perfect.
Okay, so I have already proceeded to the application. I told the initial stories. I've told about the return stories, failure economics. Okay. Is it seen?
Yes, sir, it is visible.
Could you see this slide?
Yes, sir.
Okay, fine. Sorry for the this event. And so we came to the failure opportunity you have walked out, you have talked about, and the size of the opportunity to the loss what we incur because of the failures I have explained already, and why the product and process fails we have already gone through. So we came to that general notion about why the product fails and the lack of process understanding. We have explained already the product development, process development, how it projects. Then I have explained the through a kind of solid oral example, the process options. Is it understandable? Dispersion, densification, discretization, distribution, and dosing. This is the generic term of solid flow. And the process options, unit operation options, right? Typical examples of the questions what normally are asked when a process development starts. As I'm back to the screen where I was just starting. Okay. So the traditional process development approach relied on process monitoring through static sampling, measurement, and monitoring of quality attributes and parameters in stages through certain time windows. We take samples, for example, when you are doing a blending process, we take a different position sample, but we stop the blender, take samples, analyze, and then draw certain kind of conclusion at after certain degree of blending. This is just an example. This is a traditional process development approach. And specifications were set in each stage on the assumption that all material input and process variables represent the process character in time independent pattern and on univariate relationship. This is one thing that I want to talk about, is that our traditional understanding is that the materials, once it meets the specification and passes through the controlled validated process, we get expected output meeting the expected predefined specification. This is a traditional belief. But do we know the design space of the input product, how it is processed? When we know the design space of the entire processing, in a traditional concept, somebody will say yes, but in today's modern, in 21st century's concept, it's not. We'll talk about that later on. So the specification which is staged at each stage on a static measurement, it does not really represent neither process nor the material. And the relationship, I assume univariate, the relationship. And the measurement technology were based on offline analysis, and that keeps an unrealistic time lag between the occurrence and data generation, leaving very limited possibility of understanding, communication, and control, and lack the assessment of impact of scale, equipment, geometry, and flow dynamics. That is the limitation of the process development approach on a traditional way. Now let us take the dynamic view of the process. The pharmaceutical process are mostly moving stream of materials. Can you tell me that any process which is a static process, that simply you keep the components and then you generate a process outcome? It does not happen. It's always a dynamic stream of material. For example, when the blends, when you produce the blends, it is called a blend as long as it is moving. which is, for example, when you call a finished product of a blend is never a finished product. When it goes to the either in the form of a powder as a finished product or in the form of a tablet or a capsule or something like that. So pharmaceutical blends must flow from a granulator, dryer, mill, blender to a tablet compression machine. Hence, any understanding of the state of blend needs a measurement technology which can provide real-time measurement as a function of time, even during blending and flow into the compression machine, and when the outputs are getting generated in the compression machine. Till a product is ejected out of the finished product, it is a blend. So any measurement which represents a static measurement at one point of time does not represent the true state of the product. Similarly, during a chemical reaction or a crystallization, the concentration of reactants, intermediates, impurities, products, and the various crystalline forms progressively change as a function of time, and till the completion would need an appropriate measurement technology for monitoring and control. That at a dynamic state, we are able to measure the concentration of all the components of a reaction or crystallization, and even a subsequent processing state or other purification steps. Hence, measurement technology interfacing dynamic process stream is the first step towards developing a process understanding. So measurement technology, the underlining the measurement technology word is something which we'll talk or emphasize later on. But every time that the theme of this presentation is the measurement technology. So let us understand this sequence. Measurements generate data, because a lot of misconception about that. The process data is information. Processed information generates knowledge. So information is not knowledge. Unless an information is processed, it is not a knowledge. Process knowledge with application generates understanding, right? So a knowledge, when it is not applied or and experienced, it's not an understanding. You cannot generate an understanding. The applied understanding provides control and predictive models. So that is how the measurement and control paradigm works. And this is also I'll not say it is a modern thinking. It is, of course, is a semi-modern thinking. But when you add that dimension of time, then it becomes what we call today PAT. That is called process analytical technology. The definition is a traditional ICH Q8 definition. System of designing, analyzing, controlling to timely measurement of critical quality performance attribute of raw and in-process material process with the goal of ensuring final product quality. Actually, when the PAT concept was brought out, at that time, the objective was primarily in situ measurement, precise process endpoint detection, real-time release, and continuous manufacturing. I would say that the last one is something which was the actually final goal, and other things followed. So the process analytical technology is originally manufacturing oriented technology, and it is actually took shape in the form of manufacturing technology of 21st century when the FDA, in fact, under the leadership of Dr. Azaz Hussain and his team in FDA, actually did a kind of revolutionary thinking in FDA and brought out the economics of manufacturing losses, the inadequacy of the measurement technology, the inadequacy of the process understanding and importance of that. And this is how it took shape. So general PAT scheme is as we have already indicated, is measure the key process variables about the key data and then communicate on real-time basis. The word communication and real-time is the key. Then analyze the data from the measurement to extract useful information about process conditions, and based on the convert that measurement to process understanding and knowledge, and on the basis of acquired information and process knowledge, manipulate appropriate variables to keep the process under control and maintain the product quality attributes within specification limit. And it's again an iterative process. You optimize based on the monitoring of, it is not the input or process you monitor, you also monitor output. And then input, process, output monitoring, you the knowledge and understanding you develop, then you optimize the process. So it goes on. So it's so in PAT process, it's a continuum of improvement. What are the pillars of PAT scheme? Let us understand. As we have already told, the fulcrum of PAT initiative lies in the revolution in sensor-based spectroscopic and non-spectroscopic measurement technology and near real-time sampling on dynamic, discrete, and continuous process stream. Understand this: dynamic, discrete, and continuous process stream. And the word real-time sampling, to exemplify best, is that it is the difference between the traditional sampling and process understanding and process development and modern 21st century's process understanding. The difference is between watching a movie and seeing a still photograph. When you see a still photograph, the picture you get, you continue to see that picture. By the time you understand that reality has really changed. Rather, you see in a movie or a television. So reading a newspaper and television, the experience are different. So that's how it is. And when you talk about measurement, let me clear the concept. Many people know it, the difference between at-line, on-line, and in-line. We use these terms of measurement very casually. That I have shown over here. Measurement within manufacturing environment, not online, but removing, but in the manufacturing, measurement on a scientific sampling basis at certain interval is at-line. Online is you divert the flow of the process through a device, measure, return the material into the flow. Many times it happens in API industries. And the real inline measurement, that is what we have, the ultimate measurement, where you are dipping the measurement probe in the process. So this is the difference between at-line, online, inline. We'll be using this term repeatedly during the entire presentation. Second component, the pillar, is the integrated data acquisition and process interfacing communication technology. The pattern recognition and analysis tools, along with chemometrics, have strengthened the application of PAT in gathering enhanced process understanding. We'll talk about chemometrics differently. It is a statistical tool. And it's the heart of the PAT. Without chemometrics, your spectroscopic measurement has no meaning practically, at least from the point of view of a process understanding and control. However, key is in the identification on the appropriate PAT tool that can meet the specific parameters of a process understanding need and adaptability to the process, the material characteristic, and equipment design. So what are the process analytical options? The development in sensor technology, as you have told, is in fact it is the revolutionized that entire measurement technology. The standard measurements like temperature, pH, humidity, pressure, mass, force, velocity, viscosity, and torque are well established as a part of the pressure measurement for historic period of time, and there is nothing special of it. But at the same time, it's not to be ignored. Many of the things are still considered within PAT. For example, conductivity is, we tend to understand in a low-grade measurement technology, but it's a fantastic measurement technology, inline technology, And in API industry and sometimes in formulations, conductivity measurements is the key in terms of a process control. Maybe if there is time, I will give an example on the conductivity measurement sometime. And newer application of spectroscopic involving NIR and Raman have been widely applied for metal characterization process monitoring. These are the spectroscopic tools. There's a wide variety of complex process analytical tool which is opening the possibility of a totally new manufacturing solution. And this includes acoustic measurements, acoustic sound wave based, ultrasound, electrochemical, electrostatic, optical imaging, which has reached great heights. And you would be surprised to know that today medical technology, biomedical science is primarily banking on the diagnosis and optical imaging. In fact, we are lending from medical technology and biomedical engineering this optical imaging. And what is important about all these stats that PAT is another analytical tool. The techniques that are in contact with that process via probe via probe. That is the only difference. The process in contact with the probe. And the material of the probe of construction must be considered when being used to generate process information and development mode samples, especially if that the probe is not damaged during or the process is not impacted. I'll just give a few options with a very simple measurement that's called moisture content example. Electrical capacity tomographic and electrical resistance tomography. These are primarily moisture distribution, not a single point moisture measurement. So whenever you select a method, you want to be careful about what is your need, whether you need a distribution or you need a single point measurement. So actually it's widely used in a number of agricultural fields and in some cases in some pharmaceuticals, and especially in solids and semi-solids. Microwave resonance, it works on the energy change of the reflected microwave field by water dipole, holds advantage over NIR including deep penetration depth and no requirement for chemometric model for your interpretation. NIR etc. are a little sophisticated and need a chemometric tool to really interpret. Acoustic sensor, it measures the velocity of propagation, sometimes reflection of suitable wet mass. And suitable for wet mass, not suitable will have moisture content of 15%. Maybe very wet cake when you take out of a crystallizer or before filtration, if you want to avoid the moisture content, there it may be sometimes 15%. Sometimes maybe wet granules, very wet. In wet granules, the water contains are between 20 to 28 to 30%. Where if you want to measure an acoustic sensor, may be suitable. Another moisture content example, the radio wave sensors, TDR, which has been used very much, moisture content, even for larger particles. Sometimes it is a great problem that even a sophisticated method like NIR and microwave method, if the particle size is very big, very wet particle material, they are not very accurate in terms of giving that measurement. A radio wave sensor can meet the water content, they can penetrate and give you a good understanding of the moisture content in such larger particle size situation. So primarily key is to understand what your need is. NIR sensors, of course, they are sophisticated. Primarily, as we know, in a basic spectroscopic understanding, they measure primarily OH band intensity and they are non-contact. Most of the other sensors, barring a few, many of them are contact. But advantage in NIR sensors are non-contact. No sample preparation required. Can provide chemical composition data along in the same stream for API in the product. But it has very poor depth penetration. Reliability is limited if the process material are not homogeneous distributed. Also, the multivariate data in NIR is useless without calibration against supporting references. And you need a multivariate data processing and data management and chemometric model to really interpret. So unless the process is very complex or you need really the NIR sensors one has to think of. Let us go from the moisture content model to, which are very familiar in formulation situation, is a high shear wet granulation. One which is we are very familiar is a load measurement in terms of change in power and torque. Today in every manufacturing setup, you will have a basically it is a walk measurement. The walk measurement is which to understand the granulation endpoint or the size increase. Size increase is not directly measured in that. You have to do a lot of calibration with that, the granulation size change. But ultimately, you know the weight when the actually the agglomeration starts taking, when the water bridging starts taking shape, you can roughly get an estimate of that. And a torque measurement is more of a geometric measurement, but basically they measure the two things. It is the work done, either in terms of power by measuring the kilowatt on the main or the main motor which drives the impeller, main impeller, and the measurement torque through a geometric calibration model. And you measure the torque, either of it will explain you, will give the optimum torque, will show the deflection, and the rate of change of the torque or the power consumption will show the granulation or near granulation endpoint. It is not very precise, but it's also limited by the scale dependence and the binder addition rate and the viscosity behavior of the ingredients. This is a relatively sophisticated and widely used, and it's quite used nowadays. Focus beam reflectance measurement (FBRM) is a high-speed scanning laser beam based characterization and provides a it does not make the diameter of the particle. It measures the chord length and is converted to a software, the particle size and the population trend, and it shows a very subtle change, stage-wise change in the particle size, and very accurately estimate not only the particle size and the particle elasticity also and also the granulation endpoint. The fouling is the major concern in focus beam. This is also a concern for the NIR, but I will show a kind of a case study where using NIR where this fouling has been addressed. Acoustic emission is very accurate for the endpoint detection, capable of indicating, and commercial equipments are being used, and especially in UK and Europe. This is quietly in use. It's actually measurement of the sound of a particular generated sound and a particular frequency range, and the sound pressure is measured, and the change of that acoustic emission actually tells you near the granulation change point and the end point, and also some indication about the physical property, which can be mapped and can be controlled through the chemometrics. Another thing is very useful is a drag flow force sensor. A very good tool. Is a modern tool for flow dry for both the dry and wet mass consistency measures, similar to the powder geometry approach. I'll just show the little things little later. The example of a case for a drag flow sensor measurement. What are the challenges in a high shear granulation? Let us talk a little about the high shear granulation, because I understand, at least in India, more than 70% of that dosage, solid dosage form manufacturing goes through, or the tablet manufacturing is through, and for tablet is more than 80% is through high shear granulation. So all of us are very eager to know more about the high shear wet granulation intricacies. Two major challenges. The first is the difference in equipment geometry and the process dynamics from scales to gate could complicate in the early definition of a design space. So the conclusion you derive from a design space, shall I need to explain the word design space? Somebody from the audience or the thing can tell yes or no, then I can tell what is design space. Otherwise, I believe that today everybody knows what is design space. So it is not required. to explain the design space. Okay, fine. So the early definition of design space is sometimes it has a limitation with the of the geometry and the process dynamics from scale to scale. Accurate endpoint detection, identification of the point at which the granules have reached the state is optimal, is compatible with the intended application. The endpoint can be determined by formulated as a targeted particle size mean or a particle size distribution, or it can be defined in a rheological term. But the major challenge is that granules are not really an intermediate, rarely it's a finished product. So whatever correlation of CPPs for high shear weight granulation is due to the tablet quality, finally will require to compress tablet and assessment of the CQA of tablet product or a capsule. Till that time, you will not know. So your the greatest challenge in the study, especially in the PAT studies in a high shear weight granulation, is that all measurement you do, all studies you do in the process understanding, it has to be finally linked to a compression understanding. So it is a total package. So this is the main challenge, right? So when coming back to the dynamic powder testing, these to some extent address that concern that that your understanding of the granules does not necessarily reflect your understanding of the final product. So these to some extent, this dynamic powder flow testing explains to some extent, can address this need to some extent. It is actually aims to improve the blend flowability, eliminate segregation, enhance compressibility. Looking at such influential variable, particle size density may not be, or density may not be the alone establish the robust correlation with the performance. These are all the preamble talks. It is an at-line or in-line technique that quantifies the powder flowability by measuring the axial or the rotational force acting on a blade. It is a rotating blade on a powder sample, and it works both in the granules as well in the powder. And dry or moist powder can be measured in a consolidated or moderate stress on aerated, even in the fluid bed state, can simulate the process environment and generate highly relevant data, including compressibility and permeability. So this is not only characterizes the granules and gives the process understanding of the granules, but also the compressibility picture it also gives. So finally, a direct relationship can be observed between the basic flowability energy. It measures, in fact, the flowability energy is an energy measurement, and finally, it both the dry powder as the wet mass, and even the dried granules, and finally, it reflects the compressibility behavior, right, from the extrapolating the features from a semi-dry, granules or even pellets. So this is a depiction of the drag flow sensors, drag force flow sensors. There are two, see, if I can depict, is two optical strain gauges and connected through your optical fibers. These strain gauge powder flow over it, and this creates a deflection of electrical interface. And this deflection has been found to have a quantitative relationship through an equation into the flow energy measurement. And that in fact, this flow energy can be converted into the compression behavior, particle size, particle density, flow property of the ground. So this is a very good online measurement. Initially it was at-line, but it has become, or sometimes it was a kind of a, when it was first discovered, it was a kind of a static measurement, but it is now a purely online measurement. NIS spectroscopy and granulation process. We are now coming to the spectroscopic field in terms of granulation. NIR is a rapid, non-invasive, of course, both physical and chemical properties of the material during the wet granulation process, and very useful in the PAT. It can be used for the fluid bed granulation as well, other than high shear granulation, and even blending, including estimation of the granulation endpoint, prediction of the physico-chemical product properties, particle size, moisture content. Particle density, I'm not very sure. And most important is the certain cases of polymorphic changes during the high shear granulation process can be predicted in the NIR. Of course, there are better tools in the for tracking the polymorphic changes and quantification in online things, which we'll talk about later on. But some cases, simple molecules polymorphic changes can be quantified and tracked. Here again is the profiling and sample inhomogeneity as a major obstacles. Therefore, the development of effective, robust, and in NIR monitoring method for high shear granulation process is still very challenging. This is an example. It's a kind of a case study, a kind of acousto-optical tunable filter, AOTF, NIR spectrophotometer. Here, in this case, you can read. I will explain that the NIR, it's not a probe. It is a device mounted on the discharge end of the your granulator. And acousto-optical means because there are NIRs are different kind of NIRs. There are diffuse reflectance. There are dispersive. Acousto-optical fiber is the mechanism of generation of the NIR beam, pure NIR beam. NIR, the incident light is impacted by a acoustic wave generated through a piezoelectric vibration, and it impinges at right angle to the light beam, and it causes the angular separation of the beams into NIR region. That's why it's called acoustic optical tunable filters, and these have been used. And these physical and chemical changes during granulation were acquired. Particle size, tap density, bulk density, etc., are measured, and design of experiment was conducted and evaluated through chemometrics approach. Since you have the NIR, you have to do it, and it is analyzed through a kind of a data management package. We'll talk about that. PCA and PLSR, partial least square, and principal component analysis were applied, and a kind of modeling was done. This is the case, and in this case, 11 batches of different formulations are prepared, and your 4-factor, 3-level fractional factorial design wet granulation was done, and the variables are the impeller speed, screw speed, water amount, water addition rate, wet meshing, with very common variables, and the readings were obtained, and the relationship between the particle size, hardness, disintegration were achieved, were measured, and correlations were found for all the formulations. And then the phase-wise, for each formulation, the phase-wise your pattern were matched and then mapped rather graphically. And finally, the NIR spectrum was taken. What it is necessary that you need a very because the acousto-optic has a very high scanning rate, and it actually, the granulation process is measurement based on your water measurement. It's a fast overtone wedge bond stretching is the basic, and the combination between the fundamental stretching and the deformation vibration of the wedge bond in the mid-infrared region is respectively. These are good areas where water can be monitored and quantified, and their behavior and the interaction of the particles with the water can be monitored. And that is how the changes and the optimum granulation sizing, and in this case, the spectra was shown only for the formulation 3. And there are multiple 11 formulations. Formulations only spectra was there. And you might have seen that there are two spectra, but in the IR as well as NIR, MIR, and also to some extent Raman, you need to treat, you have to give this a treatment to eliminate the many of the parameters, to the dimensionality, you have to address it. and do kind of derivatization has to be done. SNV is a standard normal variate pre-treated spectra. So that removes a lot of noise and a lot of undue, uncorrelated, relevant pseudo correlations. And that's how these spectra, you have to use always NIR through a treatment, additional treatment. And that's why the chemometrist comes in every time for NIR spectra. That is prepared. And now it is required to be explained how it is interpreted in that. The upward shift of the spectra baseline demonstrates that the particle size has increased with progression of the Higher granulation. This baseline change accounts one account of light scattering of the NIR spectra. How that is happened? Because NIR spectra, a lot of light is back reflected. It is only a very small part is absorbed. It's a lot of light is back reflected, and in back reflection, as the size increases, then there is an increase in the back reflection, and as a result, rather decrease in the back reflection, and the intensity of absorbance increases. Thus, the baseline change occurs, and that's how when the size increases, the path length changes, and that's how you can get the baseline shift in the spectra. And this in real time captured the changes in the granulation property. It is interpreted as the particle size increase. That is when your particle coalescing taking place, and then your different stages of granulation passing, particle size increasing take place, the water bridge is being forming, and then agglomeration taking place. The water is getting distributed. By measuring the water and the reflectance and the time lag and the change in intensity at the water band, you can monitor or get an interpretation of, as if you can see the particle size is changing, agglomeration is going on. And this is to be interpreted through a chemometric software. This is again a similar thing for a blending activity. Initially, the multi-components are tracked on a single rotation mode, individual components, and then for each rotation, you generate a spectra. The spectra generates for each rotation, and because you can see the sensor, the NIR sensor mounted on the blender. For big blenders, you have multiple sensors integrated to sense it. Next is the Raman spectroscopy, extensively used for monitoring of the blend, both in blender and compression machine feed system. Not only blending, but in all the downstream processing, wet granulation. An advantage that each monitors the phase transitions, polymorphic transition, solvate formation, degradation of the solvate, conversion of amorphous region, conversion to amorphous region in the granulation area, in the granulation pot. Crystallization of amorphous metal can be detected during the Raman spectra. Analysis of hydrate formation during wet granulation can be reported in the real time. After that retinaware episode, the company Abbott, as a corrective action, have installed a Raman spectra online on every, right from API manufacturing line to formulation line, every stage there is a Raman spectra probe installed, and that is all the corrective action, and that is how they have given to FDA, convinced FDA, and that is how they have been able to come back to market after little reformulating, and also these kind of installing PAT and getting a constant monitoring online and controlling the process. It gives a very tool for water solid interaction, especially in the sterilization extrusion cases, interaction with a MCC based excipients, water interaction is a good feature. I have a personal experience in handling that. The microcrystalline cellulose from two different sources for the same manufacturer, from the European plant and from the American plant. gave a different the water absorption behavior of the microcrystalline cellulose after formulation, and it was giving a different dissolution rate of the finished formulation, and the batches were failing. 100 batches failed, and before it went to market, fortunately it was within the factory, and only during dissolution testing we found that when that microcrystalline cellulose from American source of the same manufacturer, FMC Corporation, have come, and from those batches, they started failing dissolution. It was found that the water interaction with microcrystalline cellulose from that source, although all specifications are same, everything was same, and even then it was failing, because if we had a Raman spectra in the post process, we could have avoided this fiasco at the time of sterilization extrusion process online. It has been used for detection of both recrystallization of amorphous component and dehydration after tableting process. Sometimes the conversion of amorphous to crystalline to amorphous takes place during compression, during capsule filling, during blending, and this has been the most attractive mode of sensor-based non-contact real-time assay of tablet and capsule online. Tablets and capsule are coming out, and every tablet you are monitoring for content uniformity assay on real-time basis. It is like Sabri's dream without eating that plum. So you can link to that. It's our dream that every unit of the product can be tested using a PAT tool. Practical issues in spectrophotometry is actually adaptability. Both can be, fortunately today, by today's technology, they are portable, as well as there are immersion probe, process monitoring. And today, as we are telling, the process can be, the probe fouling can be addressed by putting a compressed air stream at the sapphire window of this probe, NIR and Raman probe. Small sampling area, penetration depth are limited, small effective sample volume, and sample heating, especially with Raman. Raman is an energy measurement tool. It warms up the sample, but this is neutralized to some extent. The sample stream or the product stream is moving, so the sample heating is to some extent minimized. And another challenge is adopting appropriate chemometrics. So in Raman and NIR, both have been used in for coating application. Here is an example. But both tablet and pellets coating, this is an NIR data. Most of the cases it has been used in the pan coating as well as fluidized bed coating, especially for pellet coating. What are the parameters for which, for attributes that have been measured historically and reported in the literature? From that I have taken, these are already proven, even especially in the process development application, they are being routinely used in those attribute measurement during coating, especially fluid bed coating as well as pan coating area. Most of the cases in line, some cases at line. And this is Raman spectroscopic status for coating. Coating Raman use is rather more than NIR today. You'll see in terms of in functional coating, the monitoring of or predicting drug release criteria on real-time basis and performing the curing activity after the coating. And curing activities on a polymer-based coating is very important for exact cross-linking activity of the polymer post-coating, and that is very important to monitor and link it to the dissolution rate. And here is an example in Raman spectroscopy how this has been applied and what parameters have been monitored. Inline, at-line case, most of the cases inline. This is a non-spectroscopic tool. This is an optical imaging tool, optical coherence tomography. OCT initially came in a biomedical scenario and it has been adopted, and today it is like an ultrasound with the optical imaging. It's like an ultrasound of the magnitude of the backscattered light, and along with a very microstructure of the material. Its penetration level is pretty high, and it gives a resolution more than an ultrasound to show you a spatial resolution of the material. It's not a size measurement. It gives a three-dimensional, almost near three-dimensional image, but not always three-dimensional, generally two-dimensional image, a very high resolution, and gives a cross-sectional image of the microstructure. And of course, non-contact and internal structure of the material. It has been used in a construction of a 3D semi-transparent situation or turbid material. It gives a better insight into the material. And in film-coated pellets or tablets, in a very fast-moving sample, rather in a process stream, it has been used. And I'm not aware whether it has come commercially, but at a process development level, it is being used at a very advanced stage. Hyperspectral imaging, it integrates spectroscopy and imaging to gain both spectral and spatial information. I'll give an example of how the image looks like. It creates a 3D cube representation along with a spectral representation. And the image can be viewed throughout the wavelength resolved image in a series of spatially resolved spectra, one for each point on the image. And it can determine rapidly the constituent of a solid dosage form and elucidate the arrangement of excipient versus active ingredient across a tablet. And it can be coupled with a multivariate data analysis tool. And as usual, it is designed for online. These are the designs. That left side is a hyperspectral image. You can say how the spectral information, you get the spectral image over here, and at the same time you get three-dimensional. Every plane of a cube is being seen. The entire thing is hyperspectral. And here is your OCT image. You can see, in fact, the thickness and any defect in the surface of the coating, including dimensional quantification of the thickness and any surface disintegrity or surface discontinuation can be measured, monitored, and controlled in line. And you can devise a control strategy based on your finding by interfacing with a multivariate analysis tool. Many tools are used in API processing. I can't go into the depth of everything, but typical information that we get is that the rate of product formation, the reactant loss, and the formation of impurities from side reactions or chemical degradation. And the measurements are performed during isolation will include solvent levels, particle size shape, and the polymorphic form. And particle size monitoring and characterization is also done in the downstream processing in API. These are the typical examples stage-wise. What are the instrumentation, measurement technologies used in API processing? Most of the instrumentation, as expected, is done the reaction steps and crystallization steps. And you will be seeing the FBRM has been used in the crystallization state from the particle science point of view. Conductivity has been used for especially in filtration. As you know, many of the crystallization, while doing that, sodium chloride is added sometimes to precipitate or crystallize the product to create a saturation situation, and to measure the absence of sodium chloride is to in the downstream in the filtrate is an indication of your the process completion, and where the conductivity measurement is very significant and is an online device. Drying, of course, we know moisture content measuring, mid-air, infrared region, NIR. FBRM, NIR, RM, Raman in finished product characterization. Cleaning mostly till now with monitor online in UV probes, and solvent recovery as expected is mostly gas chromatography, and sometimes cases in NIR based solvent recovery monitoring, and that too in inline situation. These are statistics in terms of giving an idea what is the extent of usage of the various measurement, the measurements in different steps in an API manufacturing. The majority, as you can see, you can expect in the crystallization and the reaction monitoring, and followed by drying, as expected. Milling is something, of course, API is, there are various kinds of milling technology used. It is necessary to understand. So the proportion of usage in process development in API scenario, it gives a picture of this. A similar picture is for formulation, for different kind of your processing technology, the use of PAT instrumentation. As expected, the major is in the RM characterization. In formulation, RM characterization is very significant, and followed by your blending. Blending is the study. In fact, in my career, I started understanding and experiencing PAT application with the blending studies in a small way in 2 of our factories when I was in PHARMA, and one is an overseas, an Israeli factory. The blending is an excellent opportunity to start with learning your PAT applications in a small scale, followed by drying and FD processing and wet granulation, high shear granulation. In fact, blending and high shear granulation today application is almost same today, and content uniformity monitoring among the finished product monitoring is also significant. Other things like coating is taking place nowadays in process development situation. Most interesting in lyophilization, while lyophilization, the use of PAT is very significant nowadays in the process development situation. Other things are, of course, corollary to that. While headspace integrity is headspace and integrity is more a factory-oriented, production-oriented technology application, but process development people are also taking interest in this. This is all about measurement. Measurement is actually only the first step in fact. And although it is, I would say 90% of, not 90, I would say 80% of PAT is the heart. But the rest of the things becomes meaningless unless you have the subsequent step. That is the data acquisition technology, data management and multivariate analysis technology, and model building package. I'll give an example of a package later on. What is most important is data acquisition and data association are the key activities, because data acquisition, for that also time is very important. All measurements are, whether it is univariate or multivariate, whether it is manually collected sample or physical samples for offline analysis in laboratory, because in a process development situation, you don't straight away go for a probe-based activity or NIR probe online activity. Just we have to do manual removal of sample or analyzing the product in our traditional way for development. That's the first step. Even for doing that, time is a very important dimension, because PAT ultimate goal is finally an FDA compliance in a regulated market. So when you do a process development, you have to keep in mind that every activity has to be time synchronized. We'll talk about that in every stage. So unless a time synchronized measurement is done, whether it is automated or it is using a PAT tool or the physical sample for offline analysis, this is a very important dimension of measurement. So let us know a little bit about chemometrics, what you talk about. The chemometrics is the next step, is actually statistical measurement, not measurement, statistical regression analysis tools, and where all maximum multivariate data analytical techniques develop to develop a model and represent a data set. Use a model to predict quantity, to predict and quantify the property of interest for new sample or process run. Because the data which comes from the measurement has both noise and signal, and all measurements are not equally significant. And these data have to be subjected to treatment, as I told, through various kind of derivatization, various kinds of segregation, the reduction of dimension of. There are so many data comes which apparently looks very correlated, and this gives a lot of pseudo correlation, and these are to be eliminated to get the really significant data, so that it is manageable and makes sense. And that is why the chemometrics treatment is required. These are all purely software. There are various techniques in chemometrics. And when the measurement takes place, no, you get the apparent measurement is the sum total of the real measurement plus noise. This noise comes from the measurement noise as well as from the process noise. So you have to have a mathematical treatment to or statistical treatment to identify those noise and remove those noise and get the real data. So there are a lot of mathematical and statistical treatments that comes through chemometrics to convert the spectroscopic data to the real data, which you really want to generate the real trend or to get the relevant information for defining the design space. Different magnitudes such as temperature, pressure, instant become comparable when typical pretreatment examples of the process data. These are the components of your chemometrics. They are the two techniques. There are many called principal component analysis and partial least square regression technique. These are the two treatment of chemometrics, which are very commonly used in pharmaceuticals, followed by the data which comes from your measurement system to treat it and make it meaningful. In large-scale data mining, the predictive modeling, especially for multivariate regression exercise, we often start large number of possible exploration predictive variables. Therefore, variable selection and dimension reduction is a major task, as I have been telling. So, it is especially for multivariate and statistical regression, these treatments are done. Often, variables use independent variables in a regression analysis displays a high degree of correlation, as I was telling, because those variables might be measuring the same characteristic, and a high degree of correlation from the predictive variables increase the variation in estimate of the regression parameters, and this is known as collinearity in the regression literature. So to address this collinearity, PCA and PLS are the statistical treatments for dimension reduction, and these are the multivariate statistical method commonly used to reduce the number of predictive variables so it is manageable and give a meaningful indication. PCA is and PLS are similar. The purpose is same, but PLS is more of an annotated level to maximize interclass variance. So many of literature you will see that a measured data is subjected to only PLS, sometimes both PCA and PLS to get a meaningful data. Main challenge in PAT is the data management is the spectral. When you are going for a PAT project, only when a process development department or R&D or manufacturing or quality assurance cannot go in the PAT project single-handedly. It has to be organizational resource consuming project. So for that, in our case, what we did, and most of the major companies would do, is the PAT team comprised of of course process development, the product development scientists. Along with that, there are quality assurance people, there are instrumentation engineers, there are chemical engineers, there are also the process engineers in that. So these teams, their interdisciplinary contribution is very important, and also of course there are your MIS people, your the so-called computer professionals, along with the regulatory professional in the team. So it is an interdisciplinary approach. And most problem, when you start a PAD project, you cannot treat any data casually, because finally it is subject to, although it is in a development stage, subject to regulatory scrutiny. And when you submit a development report to FDA, they will, if you declare that you have gone through SPAT, you have to finally get your protocol approved, even in development stage and followed by manufacturing stage. So that's why your all data, including especially the spectral data, storage arrangement has to be under regulatory compliance wave and standard. The way we casually archive the spectra or delete a spectra or replace a spectra will not do. It has to be archived in a and this needs both measurement and spectral data needs to be archived in a under regulatory frame, and with version control, with complete tracking traceability. That we'll also talk about later. Model management is also a big challenge, because the GMP, as I told earlier, the validity models has to be kept under revision control and also full traceability. And if you have a multiple sites to be deployed the same model, of course you are investing one time. So these there has to be a strategy to manage it. Method management similar to the model management, because method, when you develop a method, is very important, because chemometric method, the chemometry is used in method development. Even when you are simply measuring by a simple PAT tool, the method development, when you are especially using a spectral measurement, is very important, because you cannot use a simple calibration model. You have to use for every parameter, every project, a separate method development project, and it's very complex that way. And similar full traceability version control is required. All asynchronous data, as I told, asynchronous means what we consider that your physical measurement, and synchronized, which are directly taken on a real-time basis from the PAD, has to go from the PAD data manager, and that is a kind of a package, is a module. And when all physical sampling critical has to go there, that is why everything has to be real-time. So time is the key. Measurement of time, or keeping the dimension of time for gathering the data, although it is a physical collection of data, is very important for sampling. Multiple data sets, raw data, laboratory results are collated together in a group for PAD data management product and exported to MVA, multivariate analysis data package. And model building with the MVA package, and the model is then imported from the PAD data management product. Product means a package or a software package. It goes into this. Is a communication, two-way communication between a PAD data management package and MVA package. Multivariate analysis. By controlling and exporting of the data and import model, you have to exercise control on that. And above all, what is very important, when you are buying a PAT instrumentation, you have to be very clear and transparent with the vendor that whether his instrument is compatible with which MVA package, which your data management package. That compatibility has to be fully traceable, clearly declared, and to be established before you invest your money in that. Otherwise, land into problem. I have some kind of experience with somebody else where somebody went simply for a Bruker variety of a NIR, and then landed that the MBA package, what they are thinking suitable, does not sense from Bruker model directly. They have to go through an additional interface to really make it compatible. So compatibility between your sensing equipment, your MBA, and your data management product has to be established first of all. And of course, all these things require a full traceability. This is an example of the kind of package one I've given. The name is Unscrambler. It is widely used. It has an advantage of having for process enhanced understanding, design of experiment is handled, DOE, exploratory data analysis like PCA, cluster analysis, regression and prediction, all statistical models like partial least square regression, PLS, then PCR, principal component regression, multiple linear regression, MLR, and other treatment like derivatization, multiplicative scatter correlation, standard normal variate. You have seen an example of SNV to be prepared. Then online prediction and classification. These are the short form of online prediction and classification, OLOOP and OLOOP, and unscrambler. Like there is another as a dominant model for continuous real-time processing and quality model, and also there is a product optimizer model in the unscrambler. So when you select an unscrambler, I'm not making a promotion from unscrambler. I'm giving an example. There are many such packages. An example that when you buy a particular package of a MVA or an instrument or a data manager, then you should be able to say all your need for PATs are met with, or including your overall enhanced process understanding project. We sometimes think the PAT thing means very big in terms of hardware. This is an example of a process development laboratory setup. One side is a focus beam reflector. Suppose it's an API setup in which is a NIR, FTNIR, and you have a two reaction vessels in particle visual system. So what do you need? Reaction monitoring with an IR. You have a particle reaction control and particle or a crystallization for focus beam reflectance, and use a reaction vessels through which, and of course there are spectrometers through which you can manage your initial process development activity. But one has to keep in mind this entire data package chemometrics requirement at the end of it. So this is a very small tabletop arrangement of a process development lab. So if somebody thinks PAT means for a full building, it's not like that. You have to, a part of a lab can be converted to a PAT lab to start with, but you should have all the instrumentation. You're compatible with what you are going to scale up in a process development lab, in a factory setup. So that you have to keep in line, so that there has to be a clear symmetry in that. Any asymmetry can jeopardize your PAT project. This is just a statement. The difference between the PAT in process development from a manufacturing setup. In development, you finally, the reliable measurements, what is the intention? Reveal the previously unknown process, components, mechanism, and relationship between variables, and leading to development of predictive models, both mechanistic and chemometric. And predictive models can finally, qualitatively or quantitatively, work of the develop. As the development progresses, you can map the design space. And finally, we want to develop a control strategy out of the definition of the design space. And here, this is a listing of, as a summary of the process development, what are the objectives, what we really do, and in manufacturing, what are the objectives for a PAT. The process development, always keep in mind, is a process understanding. Whereas in manufacturing, it is a control, monitor and control, right? And of course, trend analysis, continuous process verifications are the order of the day. And you need essentially multi-component analyzer, because you must be doing a multiple component at a time to analyzing at some time. But when you identify what is your target, then you can go the target component analyzer in the manufacturing stage. Data complexity is much more. You essentially have to go for a multivariate analytical tool. In factory situation, unless you are using a spectroscopic PAT tool, you can still manage with a univariate in certain cases, especially in API situation. Support requirement, you need a very high level in the process development, a high level expertise and a continuous support, sometimes physical sense of the expert. Whereas in manufacturing situation, you can remotely the expert can advise you. The quality system, you have to, although it works in a development, but with the full traceability and version control with change history documentation. Whereas in a manufacturing, of course, full GMP. PAT expertise primarily required in the method design and development. Method development is the key in the PAT measurement. And whereas in expertise in manufacturing is operation and maintenance. Right. So enhanced process understanding is something which I want to just in one slide finish off, because that is the key, that is the objective. Only measurement and then controlling a process is not the understanding a process. The process is most important, and it starts with the definition of a design space, knowing the boundaries, knowing the equations between the variables and the both independent and dependent variables and the outcomes, and in the form of equation, maybe linear, maybe in terms of quadratic, and maybe logarithmic, the relationship. It may be an empirical formula, empirical relationship which can be worked out, but there is a need for definition of a quantitative relationship to generate a model. And once a model has been developed, it can be used to further optimize the process. Important is selecting set points, evaluate the design space, establish the limits, both on the inputs and outputs from the process, and develop with very underlying statement to derive a control strategy. Unless a control strategy is derived from a simple control strategy. Suppose you are milling a product. In a mill, as per our today's knowledge, what changes, what controls the particle size or distribution? Maybe the screw speed, feeding speed, impact speed, if it is an impact mill, primarily for a given your sieve plate, because sieve geometry, plate geometry impacts, but for a given sieve plate geometry, these two things impact. So what you can change to control the particle size is the feeding rate and the screw, that is the screw feed rate, or the your impactor speed, right? Maybe the design of the impactor speed. So these are the control strategy. How you control? But if you get a monitor the output particle size distribution through a probe, there has to be a feedback mechanism to go to a controller which senses and interprets the output need and manipulates the feed rate as well as the screw speed or the impactor speed. And that is how this is the control strategy. How you want, this is a very simplest example of control strategy, how impact. And for any process, there has to be a control strategy. So only knowing is not important, that you know the particle size distribution. And you spend thousands of hours to think of how to control the particle size. So working out a control strategy is the ultimate goal of a PAT, process development activity. And this is my favorite slide. Thank you very much.
Thank you, sir. Thank you for your outstanding presentation on process analytical technology, PAT technology application in process development in pharmaceutical industry. Now I would like to hand over Professor Jayanta Chattopadhyay to sum up the talk of Mr. Sampad Bhattacharya and convey the concluding remarks. Professor Chatterjee, please.
Okay, Dr. Hossain, why do you give such difficult task to me? You know, this is my complaint. You know, summarizing this kind of, you know.
Would you kindly unmute your video? Turn on your video.
Okay, let's see. 1 minute, please. Once again, my video is not working well, but then again, give us a time to cool down for.
Sure.
After this talk, you need some time.
Yeah. Yeah, you are fine.
Okay.
You're fine now.
Okay. So, let me be a little bit of nostalgic, Sampada. I listened to you in 1976, almost 44 years. Okay. So you are, explaining to your classmates in pharmacy department library, and Remington was open in front of you. Maybe you are in the final year that time. And that time he was crisp and clear, very crisp and clear. And he was explaining to his classmates, it is a kind of lecture that what we heard today. And after 44 years, maybe we are listening him again, and he is crisp and clear. Okay, now let me start my task. Sampath Bhattacharya started with, the failure economics. The loss due to quality failure, even in small companies, every, after a few batches, we come up with a failure batch. So this failure is very common, and there is a cost, and he started with failure economics, and then, he started how to prevent it, the quality failure. And then he mentioned that there is a big opportunity, some $1 billion opportunity in preventing quality failure globally. Of course, there is a big opportunity, and how Pat came in the picture. And he talked about the contribution of Dr. Hossain. And then he switched over to process development. How during process development, this Pat can be utilized, process analytical technology. And then he started, particularly, PAT is a vast area, so he started chosen one area, which is a very difficult area, that is oral solid dosage design, where, there is a solid state physics, and he started on that area, and how the sensing can give us a wealth of information, what is happening during granulation process where solids are interacting. And particularly in granulation, he explained the granulation process. I cannot remember every bit of it, but how the granulation happens, and the compactness happens, and how the agglomeration happens, and all this, and how process analytical technology can help us to understand what is going on, what is the physics going on inside agglomeration, and the force and the viscosity and the measurements. Then again, coming to the quality attributes, critical quality parameters, understanding. And then he talked about many things. It's very difficult to mention everything. He talked about blending. I remember FDA once, put up a guideline on blending, and they quickly withdraw this, blending guideline. Industry people jumped on FDA. It was a draft guideline, but blending is a difficult subject, and particularly when the blender size goes high, I mean to say something around 5,000 liter or 10,000 liter, and solids are mixing, and solid state mixing can be a mixing, demixing cycle, and all these complex things happens in solid. It is not a liquid mixing. So unless we have a high-tech, process, high-tech technology, in the sense that, you can have sensors to understand what is the force, and what is the, what is the viscosity, what is the particle size changes happening. So a lot of things came in from, I believe, from the chemical process industry, where FDA was not there. So naturally, they are free to do whatever they want, or they are free to put all science. But then PAT has to take the challenge of FDA, because FDA, whatever you do, it has to be done as per, analytical validation, and the regulators will, jump on you and examine every document, every data, and data integrity and everything. So then, Sampath Bhattacharya talked about the dynamics of the process, static sampling versus dynamic sampling, which happens, once we are moving from a batch operation. Classically, even today, we are morely a batch manufacturing process. Pharmaceutical sciences essentially was a batch manufacturing process, and from then, once we are entering into the era of continuous manufacturing, then data acquisition and dynamic data acquisition becomes very, very critical. He mentioned that, and the management of measurement technology. The developments in the measurement technology is becoming a very hot topic today, and a lot of advancement can happen. He talked about a lot of instruments, the measurement technology. I remember in 92, when I joined Pfizer, the first thing I came there, that they have a NIR group. I'm talking about 92, many years ago. In 1992 or 93, they have developed a NIR group, and they are planning to put NIR in every facility so that they can check the raw material quality at the gate. So remember, and we struggled with that, the software and chemometrics in those days. We could not understand much in those days. But then again, from 92 to 2020-21, NIR and Raman, as Sampath Bhattacharya mentioned again and again, NIR and Raman, these two technologies becomes very critical in raw material identification and process monitoring, and in all sort of technologies, formulation technology. These two are, I believe, they are very critical. Then coming down to other issues, like once again in PAT, in design space, and in process development. If we really have to do a process design as per the current FDA way, we have to define the design space, and we have to explore, and so that our knowledge space increases. That means we have more knowledge on the process itself. Enhancement of process knowledge, that is the term he used. It is very critical that our knowledge of the process, more knowledge we need to gain about the process, then only we can solve problems. These failures, whatever happens in day-to-day life in the pharmaceutical processing, one of the reasons may be due to our lack of understanding of the basic science behind whatever we are processing. So that knowledge gaining is very important, and that can be done through this process analytical technology. We can have a dynamic acquisition of data, what is happening during either in solid phase or in other processes. So in IR, once again, I am coming to NIR and Raman, he mentioned about, and then he will mention about a lot of new technologies. Now I understand from him that our pharmaceutical science has become a rocket science. Earlier we used to have a very simple things. Maximum we can have a strong and cob, tablet hardness tester, or a simple tablet hardness tester, Monsanto. But now from that simple hardness tester, now we have gone to this kind of focus beam scanning equipment and laser probes. Now he talked about in one slide, I am not following the sequence. What a PAT lab should have as the API is concerned. So there is a laser probe for particle size distribution analysis, and in a small two vessels or kind of reactors, where you can measure during crystallization, you can have a particle size distribution measurement. Then you can have a FTNIR or NIR or some kind of Raman equipment, so that you can, during your crystallization, you can track your polymorphs, you can track your particle size distribution, and you can do a lot of experiments, so that crystallization experiments, so that you can understand what kind of polymorphs is forming in different solvent ratios. So that gives an enormous opportunity for patenting, and in a very small amount of time, one can do a lot of experiments. And coming back once again to his emphasis on optical coherence tomography. The other day we were discussing, our Honorable Vice Chancellor, during our meeting has given us a problem. He said that just at looking at a tablet, he will take out a tablet from a strip and ask us, can you tell us whether this is adulterated or not? You develop something like that. So people started suggesting I kept mum, because I know, because in the core, if there is any impurity in the core of the tablet, how do I detect it? Simply I started thinking, can I do a CT scan or some MRI? Like I will go to a scan center and ask him to do me a MRI. Now I understand from Sampath Bhattacharya there is a optical coherence tomography, OCT is happening. It is really happening. and hyperspectral imaging. Hyperspectral imaging. So what is happening inside the tablet also? We can have a 3D scan, and we can have a spectral, observance. And we have become like kind of astronomers. By sitting in this earth, we can know what is happening in sun. So sitting here, we can know what is happening inside a tablet, inside the tablet coating, by this optical imaging, hyperspectral imaging, and optical coherence tomography. Difficult to spell also. Difficult to spell also. So naturally, definitely, you can understand these technologies are very sophisticated, and of course expensive. And I believe it is expensive. But then again, coming back to the money one can save, end of the day, I once again go back to his initial statement that when, end of the day, it is all capitalism. That means I can develop a very sophisticated and very advanced machine, but unless it can reduce some cost somewhere, then that technology is not probably will not going to be sustainable. So naturally, those who are investing in PAT, there might be some benefit in terms of cost saving, in terms of failure reduction. So then again, going to the chemometrics, he talked about chemometrics. Definitely, there are, going into the depth is definitely beyond my scope. Then chemometrics, then principal component analysis, then multiple regression analysis, and understanding chemometrics is quite difficult. I would request my young faculty colleagues that hold questions for the moment today. I would request you to read this presentation and read on this PAT first, and then you can come back with the questions. Because unless we go a little bit of depth, there is not much point of asking questions. Let us have some idea about this subject, and then finally we discussed about something. He talked about unscrambled labs like PCA, principal component analysis, because there is a big data this PAT is collecting, and understanding this big data, we really understand, need some tools to have some statistical kind of analysis, that principal component analysis or principle of least squares or multiple regression analysis, and all these are particle vision microscopy, that is also a tool in particularly in API development. At some point of time, we pointed to the management that we need so many tools and so many all these things. But then again, all these capex proposals are usually scrutinized, and they said, okay, no, next year, you are this year you are asking for too much money. So PAT has with time, this enhanced process for the purpose of enhanced process understanding, and particularly to explore the design space and the knowledge space, and finally to develop a control strategy. End of the day, we need to control our process so that failure doesn't happen. PAT is definitely a real advancement in this field. So with this, I thank Mr. Bhattacharya for such a excellent lucid overview of the subject. And I would request Dr. Hossain and Dr. Pranavesh Chakraborty to take over. And as far as I am concerned, it is a big learning for me to understand what the world has, where the formulation development world or the pharmaceutical field has gone. Regarding question answer session, I know if I allow these questions to come in, there will be a big flow of questions. I don't think we will we will cross midnight today. So I will request my young faculty members to hold, put your questions, and maybe in the last day, when we'll have the panel discussions, then you can put your questions by email or something. We can handle those questions. With this, once again, I am thanking Mr. Bhattacharya for such an excellent presentation. Thank you.
Thank you, Professor Chattopadhyay, for your quality discussion on the topic presented by Mr. Sampath Bhattacharya. Yeah, so participants can note their question, and they ask their question in the last day of the program and during the panel discussion. I think it would be more convenient. Now, I would like to request our Director Sir, Professor Pranabes Chakraborty, to say a few lines to close the session today. Sir, please.
সবার জন্য শুভ রাত্রি, আমার জন্য শুভ সন্ধ্যা। আমি একটু বাংলায় বলি, অনেকক্ষণ ইংরেজি শুনলাম। সেটা হচ্ছে যে আমাদের যে সমস্ত লার্নেড ফ্যাকাল্টি মেম্বার্স সম্পদ ভট্টাচার্যের বক্তব্য শুনলেন, আমার মনে হচ্ছিল এটা যদি আমরা ইউনিভার্সিটিতে অফলাইনে 7 দিনের বা 10 দিনের একটা ক্লাস করতে পারতাম, তাহলে বোধহয় আমাদের সবারই অনেক উপকার হতো। সম্পদ আমাদের মধ্যে সত্যিই সম্পদ। তোমাদের একটা কথা বলে রাখি, একাডেমিক্যালি, ইন্ডাস্ট্রিয়ালি যেমন ইকুইপড, সম্পদ ভট্টাচার্য is a name and fame in pharmaceutical world, but beforehand he is one of my closest friend. And তোমাদের একটা ইনফরমেশন দিই, সম্পদ কেন সম্পদ। সম্পদ যে 7 দিনের বা 10 দিনের ক্লাসটা আজকে সুন্দরভাবে সবার সামনে তুলে ধরলো এবং ইউনিক ম্যানার, অপূর্ব তার বিশ্লেষণী ক্ষমতা, এবং এই সম্পদ একটা সময় আমরা কোনো ফাংশন ভাবতে পারতাম না যে সম্পদ কোনো না কোনো জিনিস বাজাচ্ছে। সম্পদ ভট্টাচার্য সমস্ত রকমের স্ট্রিং ইনস্ট্রুমেন্ট এবং চামড়ার ইনস্ট্রুমেন্ট অসাধারণ বাজাতো, এবং ও যদি ওই লাইনটা তো এগিয়ে আসতো, আমার মনে হয় ভারতবর্ষের একজন শ্রেষ্ঠ মিউজিশিয়ান হিসাবেও এবং ম্যাস্ট্রো হিসাবেও নাম করত। ও এত কাজের ফাঁকেও এই চর্চাটা চালিয়ে যায়। এখন অবশ্য ফাংশন করে না, আমার সঙ্গে কথা হচ্ছিল, এখন কিছু লোককে শেখায়, আবার অনেক সময় আমাদের এখন তো ইউটিউব বেরিয়ে গেছে, সেখানে দিয়ে ছাত্রছাত্রীদের শেখায়। তো অনেকদিন পরে সম্পদের সঙ্গে আবার ফোনে তো আর এত কথা হয় না, এই সাবজেক্টটা যখন আমাকে সম্পদ ওর পিপিটিটা পাঠালো, আমি সঙ্গে সঙ্গে দেখে বললাম যে ডক্টর হোসেন, এইটাকে পুরোপুরি সারসংকলন করার জন্যে আমাদের মধ্যে একজন উপস্থিত আছেন, যিনিও কিন্তু ইন্ডাস্ট্রি ফার্মাসিউটিক্যাল ওয়ার্ল্ডে একজন আমাদের মধ্যে কিংবদন্তি। সুতরাং সম্পদ নিয়েছে 7টা ক্লাস, আর জয়ন্ত স্যার নিয়েছেন 2টো ক্লাস। দুটিয়ে আমরা কিন্তু আজ 9খানা ক্লাস করলাম।
জয়ন্ত স্যার একটা ডাউট ক্লিয়ারিং ক্লাস হয়ে গেছে।
এবং সব ব্যাপারেই যেটা হয় যে কি করছি, কেন করছি, কিভাবে করছি, কি জন্য করছি বলার পরেও কিছু প্রশ্ন থেকে যায়। সেই প্রশ্নগুলো আজকে সামনে তুলে এনে ধরেছেন জয়ন্ত চ্যাটার্জি, কারণ তার যে ব্রড স্পেকট্রাম আইডিয়া এবং নলেজ অ্যাবাউট দ্য সাবজেক্ট, সেটা আমরা সবাই শুনলাম। সম্পদকে অজস্র ধন্যবাদ এবং অকৃত্রিম ভালোবাসা, জয়ন্তকেও তাই। ডক্টর হোসেন, আমার মনে হয় যেটা জয়ন্ত বলেছেন যে প্যানেল ডিসকাশনের দিন মূলত প্রশ্ন যেগুলো ব্রড প্রশ্ন সেগুলোকে করার, কিছু কিছু প্রশ্ন আজকে কিন্তু আমার মনে হয়, আমি এখানে দেখলাম 6 জন 7 জন হাত তুলেছেন, কয়েকটা প্রশ্নের যদি সরাসরি সম্পদ আজকে খুব বেশি নয় গোটা 5-6 প্রশ্নের উত্তর দিতে পারেন, তাহলে আজকের এই সম্পূর্ণ ওয়েবিনারটা প্রচন্ড সাফল্যমণ্ডিত হবে। এবং সেক্ষেত্রে আমি সম্পদকেও অনুরোধ করব যে ডিটেল ডিসকাশন প্যানেল ডিসকাশনে হবে, কেন সেখানে ছাত্ররাও তো থাকবে। আজকে কিছু প্রফেসর তাদের ডাউট ক্লিয়ারেন্সের জন্য বলেছেন, আমার মনে হয় সেগুলো 2-4টে প্রশ্নের উত্তর দেওয়া, পরবর্তীকালে সবাই আমাদের মেলে পাঠাবেন, আমরা একটা মেল পাঠাবো, সেখানে আমরা সমস্ত প্রশ্নগুলোকে ঝাড়াই বাছাই করে প্যানেল ডিসকাশনের দিন দাঁড় করাবো। এই বলে প্রত্যেককে, সম্পদ ভট্টাচার্য, ডক্টর চ্যাটার্জি, ডক্টর হুসেন, আমাদের এই পর্দার অন্তরালে থেকে প্রেম কুমার যে অসাধারণ কাজটা করে যাচ্ছেন, রাউন্ড দ্য ক্লক, ভোর থেকে মধ্যরাত্রি পর্যন্ত কন্টিনিউয়াসলি যোগাযোগ রাখছেন, এবং আমাদের যে সমস্ত শিক্ষক শিক্ষিকা, ইন্ডাস্ট্রির যে সমস্ত মানুষেরা এসছেন, তারা নিশ্চয়ই এই যে সেমিনার চলছে, ওয়েবিনার চলছে, তার থেকে উপকৃত হচ্ছেন। আমরা আগামী দিনেও এরকম অনেকগুলো এফডিপি করবার জন্য মনস্থ করেছি বিভিন্ন সাবজেক্টের উপরে, এবং আশা করব সম্পদ ভট্টাচার্য আজকাল কলকাতায় খুব কম আসে, যখনই আসবে যদি ফিজিক্যাল মোডে কোনো ক্লাস হয়, আমরা চাইবো আমাদের ইউনিভার্সিটিতে এসে সম্পদ ভট্টাচার্য আমাদের ছাত্রছাত্রী এবং শিক্ষক শিক্ষিকাদের সামনে যে কোনো সাবজেক্ট, তার যেগুলো এক্সপার্টাইজ রয়েছে, ওর অবশ্য সব ব্যাপারেই এক্সপার্টাইজ আছে, সেগুলো নিয়ে যদি আমাদের খানিকটা সময় দেন, আমরা খুবই উপকৃত হব। এই বলে সবাইকে শুভ সন্ধ্যা বা শুভরাত্রি জানিয়ে আমি শেষ করছি। ওভার টু ডক্টর মাসুদ।
থ্যাংক ইউ স্যার, থ্যাংক ইউ সো মাচ স্যার। তা আমি তাহলে 2-3টে রিপ্রেজেন্টেটিভ কোশ্চেন সিলেক্ট করে নিচ্ছি, যেগুলো মানে অলমোস্ট বেশিরভাগই কোশ্চেনকে কভার করে দেবে। একটা কোশ্চেন, what are the things a faculty should percolate to a graduate coming to industry? What are good source material and reference for better understanding?
Okay, this is a very broad question. If you are meant to say an undergraduate means just somebody doing B.Pharm into industry?
Yes.
So it depends on, say, the industry is a very big area. I mean, there are a lot of department divisions and all. Somebody may make career in the manufacturing, somebody may make career in quality, somebody may make career in other areas. Somebody may make quality control, somebody may make quality assurance. What is very important is that, so that's why a simple recipe may not be all. But single recipe which is very important is that make them strong in fundamentals. If somebody, generally in India, most of the people make career in solid oral and also in injectable. These are the two broad areas in manufacturing. So fundamental principles of operation, for example, if they know the unit operations, if somebody is going to come to manufacturing, unit operations, understanding unit operations, their principles is very important. And if somebody is going to make a career in API, there are basic fundamental things what we reach in our pharmacy curriculum is not bad, but some kind of understanding of the current regulated environment is today's requirement, because the way you have gone through your academic environment, and when you go to the manufacturing environment, you have to put yourself in a disciplined regulatory operation. For example, you cannot how to document, how to read document, how to do documentation, how to archive documents is a requirement, other than the fundamentals of science what we reach in our normal pharmacy curriculum. What is being taught is good enough. Only thing is that keeping a track of the current guidelines. For example, we have been talking about blend uniformity, but if somebody has gone through, although the guideline has been withdrawn, there are a lot of things to learn about the limitation of blend uniformity. Similar things, you keep a track on the guidelines. I'm not talking about a very sophisticated subject. Things like your blend uniformity is one of the issue. Then there are cleaning, the concept of cleaning, batch cleaning, cleaning validation. Those kind of things are relevant today if somebody is going to manufacturing. If somebody is going to quality, then is more depth in terms of understanding the principle of dosage form, the principle of variability. And one thing which is not being taught in, I believe, in the academics is the quality science. The quality science which is being taught in pharmacy curriculum is generally your typical quality control subject, and or how to do an analysis or how to do a sampling or those kind of things. But what is basics? I don't know to what extent a basic theory of something is being taught. How much statistics is being taught, and these teaching the statistics, teaching the process control are very important. So making them aware and understanding the process control science is key to for any manufacturing as well as quality assurance person who want to build career. I can talk on this for maybe 2 hours. So I think I must restrict myself to this.
Okay, sir. Thank you. Another question: is there any special statistical package is using by industry for uni or multivariate calculations,
There are many.
or regression analysis like SPSS that are nowadays commonly using by research scholar for their statistical statement?
There are many. I don't want to take a name, but there are plenty. You simply search net, you will get plenty. Industry use a different kind of statistical package. Yes, and they don't buy a single statistical package. Means there is a part of the overall quality system package. In that there will be statistical package. Like for example, TrackWise. TrackWise is very common, and it addresses both your quality system primarily and also take from the quality control system. TrackWise has all built-in quality system related documentation packages.
Okay. So another question, Dr. Madhusudan Ghosh. I think you know him, Dr. Madhusudan Ghosh. He said that, dear Mr. Bhattacharya, I enjoyed a lot by your narration of very interesting topic. But my question is something else. How much India has progressing in manufacturing the synthetic drugs or basic raw material? Please do remember in 1978 IPC at Calcutta, JU, when one article was written by Dr. Ghosh and edited by, I think, you, a brief review of development of synthetic drugs. Even after 4 decades, that very status of India in manufacturing raw material has bound me.
My simple answer to this will be, for synthetic drugs, India has progressed a lot. And only problem is that we start from very advanced intermediates. And most of the advanced intermediates used to come from China. And the major reason for not doing so far in India was the pollution control. And China, as you know, they're liberal in terms of that area. I don't want to talk much on this. But now government of India is reconsidering that barrier to some extent. They are trying to address the issue. And in that case, we will be able to start manufacturing many hazardous intermediates. Many of the intermediates manufacturing, or rather key intermediates, are very hazardous processes. We don't want to go as a country at the cost of environment. That's the basically writing. We are fairly disciplined country. That is one of the bottleneck. Otherwise, so far as knowledge and technology is concerned, I think is not a barrier. And another third element is the costing. Of course, compared to the advanced world where you are very much competitive, but when you go to China, we get it, we are impacted very adversely because they give at a inexplicable low price, which does not even cover the material cost. That is difficult to compete. Probably they don't have any cost incurred on infrastructure or in the pollution control area in control discipline. So this is the basic bottleneck. Otherwise, I have no doubt in terms of capability, there is no issue, and we can manufacture any drug. Only to manage the cost. Technical capability is wise, no issue.
Thank you, sir. Thank you. These are all the major questions.
Okay.
So thank you very much